Hi, my name is Cemre Kefeli, and I am a master's student in Istanbul University of Molecular Biology and Genetics. Today, I will talk to you about our quest of identifying novel targets in Wnt signaling pathway. Wnt is an evolutionarily concerned pathway which controls development of the organism, and it is associated with various types of cancer and Alzheimer's disease. In a normal cell that is not stimulated by a Wnt ligand, beta-catenin is degraded by the destruction complex. Beta-catenin levels kept low in cytosol. When a Wnt signal is present, the destruction complex is held and the beta-catenin accumulated in the cytosol. The beta-catenins travel then to the nucleus to interact with the TCF left transcription factors. That leads to the transcriptional regulation of the Wnt target genes. Genes that possess a TCF left binding site in their promoters are called direct target gene. Some of the binding sites are not functional, so we need more information to identify a target gene. The first target gene discovered in 1998 after the emergence of the key Wnt signaling components, and the number of target genes grew over time, and we think that there still might be a target genes that are yet to be found. We hypothesize that target genes behave kind of similarly. Not identical, but in a similar fashion. So we thought if they behave similarly, that might be seen in the gene expression values of target genes. Thus, we used gene expression values to train a machine learning classifier that might see this similar behavior. Following several studies, we assumed that target genes may be tissue-specific. We decided to focus on colorectal cancer, and we collected the data according to this assumption. Choice for colorectal cancer was due to the abundance of data in this tissue compared to the other tissues or diseases. We used this data to train a special kind of machine learning classifiers. What classifiers basically do is separate data into groups. After training, it can recognize Dali from Van Gogh. This is basically how supervised machine learning works. The positive and negative labeling denotes the binary nature of the problem. It does not refer to an inherent value. We show the classifier the negative and positive examples during training. After training, the trained classifier can assign a label to new cases. Machines do not see the pictures, or in our case, the genes, as we do. We have to represent them in a categorical or numerical values, which is a more machine-friendly way. We represented genes with differential expression data, which is a collection of genes represented as numerical vectors. Our goal is to identify novel target genes in Wnt signaling pathway. Contrary to classical machine learning problems, we only have validated positive cases, also known as bona fide target gene. We do not have a validated non-target genes of the pathway. So we have some positive cases and no negative cases. Since there might be other target genes we yet to know, our set of bona fide genes is also incomplete. Hypothesizing that the most of the genes are not target, the rest of the genes, all genes except the bona fide targets, would mostly contain the negatives but some target genes. This resembles to discovering a new painting of a famous painter. How will we decide if it's fake or not? We have a bunch of paintings at hand, in this case, genes with their target state is unknown, and we have to identify genuine piece among them, which is in our case, it's the target gene. For the positive examples, we compiled a list of genes validated as targets. Hypothesizing that most of the genes are not targets, we chose negative examples from the rest of the genes. We used separated positive and negative cases to train the classifiers. After the training, we used the training classifiers to assign a label to each gene. We counted the number of times each gene is classified as target, and we calculated score based on this voting system. Classifiers are different from each other because every time we train, we use a new random set of negative examples. If the majority of votes are target, we deem the gene as target. We need to do some tinkering while training. Classification depends on the parameters called hyperparameter, which essentially affects the performance of the training. Our hyperparameters are sample size and penalty. Sample size is the number of randomly chosen negative examples in each training, and we applied penalization for the misclassification of the genes. We also trained classifiers with fake positive targets, which are chosen randomly. This is our negative control. We did this to validate that our compiled genes have a shared characteristics, and randomly selected genes cannot produce the same results. In the end, we trained two sets of classifiers. First, with bona fide targets and randomly chosen negatives. The second, fake positives and negatives, both also chosen randomly. We used several sample sizes and penalty to see which one works the best. After the prediction, we compared these two sets to see their performances. The red numbers show the novel genes predicted by classifiers trained with bona fide examples. Blue numbers show the novel genes predicted by classifiers trained with fake positives. Recall is the rate of recovery of the positive examples. Since we do not want too many novel targets, our best results in hyperparameters are size 1200 and penalty 10. We recover all bona fide genes and predicted 108 novel target gene candidates. The genes that have the highest score are patched1 and the GLE3. The interesting thing is these genes have important roles in hedgehog signaling, so this might suggest that there might be a crosstalk between WNT and hedgehog signaling. In parallel to our story, Boonkamp et al. Also recently proved that patch1 is a colon-specific WNT target. We also predicted these genes as targets, and these genes have been reported as WNT targets in tissues other than colorectal cancer. What are key takeaways? We can find good candidates for experimental validation. Some of our predictions have already been validated. Also, when target genes share some similar expression patterns, and the machine learning method can discover new targets based on partial knowledge of positive examples. This was our study, and thank you for listening.